Imagine like a burglar breaks into your house, but instead of just hiding, they completely rewire your smart home system to, well, lock out the police and order themselves room service.
Yeah, that is exactly what the tuberculosis bacteria is doing inside our immune cells, which is honestly terrifying.
It really is. So welcome to the OriGene Technologies podcast, and we are so glad you are joining us for today's deep dive.
Absolutely.
Our mission today is to explore this really groundbreaking 2026 Nature Communications paper that exposes exactly how this microscopic heist works. And we are also going to highlight how OriGene Technologies is incredibly proud to support this vital research through their products.
Right, and it's a genuinely fascinating study because when we think about Mycobacterium tuberculosis, or, MTB, we usually picture a pathogen that's just trying to survive inside our macrophages.
Right, which are the very immune cells dispatched to destroy it in the first place.
Exactly. But this research proves it isn't just passively surviving, it is actively reprogramming our cellular lipid metabolism so it can thrive.
Okay, let's unpack this. Because macrophages are literally built to destroy bacteria. How does releasing like one basic enzyme overpower the immune cell's entire defense system?
Well, the mechanism is brilliantly deceptive. So once inside the macrophage, the bacteria releases this enzyme called isocitrate dehydrogenase, or IDH.
Okay, but normally IDH is just a basic metabolic enzyme, right?
It is, yeah. But inside our cells, this bacterial IDH physically latches onto a critical host nuclear receptor called PPARγ.
Oh, wait, I know PPARγ regulates lipid homeostasis, right? Like managing fats in our cells.
Spot on, and here is where it gets really interesting. When the bacterial IDH binds to PPARγ, it physically blocks the normal degradation of that receptor.
Oh, so it just builds up.
Exactly. It acts like a shield over this precise location called the S112 site, which prevents phosphorylation. So because the host cell can no longer tag PPARγ to, throw it in the cellular trash can, you get this massive, unnatural buildup inside the macrophage.
Wait, so what does this all mean? Why does the bacteria actually want to hoard all this PPARγ?
Well, it triggers this deadly domino effect. The bacteria uses all that excess PPARγ to recruit a specific host protein complex called NCOR SMRT.
Okay, NCOR SMRT. And what does that do?
You can think of this complex as a massive off switch for DNA. Together, they travel to the host's DNA and physically shut down the transcription of a really crucial gene called GPIF4.
I mean, I'm completely lost. What does GPI4 normally do for the cell?
So the protein it produces, GPX4, is basically the cell's bodyguard against a specific type of destruction called ferroptosis.
Ferroptosis, that's an iron-dependent form of cell death, right? Caused by catastrophic damage to lipids.
You got it. By shutting down GPX4, the bacteria strips away the macrophage's bodyguard. The macrophage membrane suffers massive lipid damage and dies via ferroptosis.
Oh, wow. So it literally bursts open, allowing the bacteria to persist and spread. That is molecular sabotage on just an insane scale.
It really is.
But seeing something that small, like a bacterial enzyme hugging a host receptor to block a single phosphorylation site, I mean, that's virtually impossible with standard tools.
Yeah.
How did they actually prove this was happening?
Right, they couldn't just guess. They needed absolute proof. To verify that IDH and PPARγ physically interact, the researchers engineered a special version of the bacterial IDH.
Oh, did they tag it with something?
Yeah, they tagged it with a fluorescent protein called zesgreen, which let them track the bacterial enzyme's exact movements.
But even with a glowing tag, pulling interacting proteins out of a crowded cell must be incredibly difficult.
Oh, it's notoriously tricky. They used complex techniques like co-immunoprecipitation and Western blots, which is essentially fishing out the proteins to see if they are physically stuck together.
Right, and when you're dealing with host-pathogen interactions this complex, you need an antibody that binds perfectly to your target and absolutely nothing else.
Exactly. And that is why the researchers relied on OriGene Technologies' highly specific mouse monoclonal zesgreen-1 antibody.
Oh, that's a clone of TI2C2, right? SKU TA1800002.
That's the one. That specific clone gave them the exact specificity needed to pull down the zesgreen tagged IDH and prove, without a doubt, that it was binding to the host's PPARγ.
That's incredible. It really highlights how essential, reliable tools like TrueMab OriGene antibodies are for researchers dissecting these complex interactions.
Yeah, the data is only as good as the tools used to capture it. And if we connect this to the bigger picture, Understanding this IDH-PPAR gamma axis is just a game changer for you and the broader medical community.
Because it opens doors for new host-directed TB therapies, right?
Exactly. Instead of just trying to kill the bacteria with antibiotics, we could potentially use PPARγ gamma antagonists to block this interaction, save our macrophages from ferroptosis, and let the immune system do its job.
Discoveries like that are exactly why OriGene Technologies is so excited to be part of this research, providing the tools that make these breakthroughs possible. So we'll leave you with a final thought to mull over today.
Yeah, this is the part that really gets me thinking.
Right. If a basic bacterial metabolic enzyme like IDH can secretly moonlight as a master genetic hacker in human immune cells, what other basic bacterial proteins are quietly orchestrating human biology right under our noses?